Now, in continuation to the previous aspects, the most important concept in biochemistry, when you are studying this biochemistry, you should consider is the bioenergetics. Because what we are saying that the cell is carrying out its metabolic activity solely for the first and prime purpose to sustain itself, to remain living. And the first and the foremost requirement for sustenance or survival would be availability of energy so that the cell is capable to provide for the energy required for carrying out the biochemical reactions. We know that this energy is obtained through various different metabolic reactions going on inside the cells. Basically, all the chemical reactions they have been categorized as or categorized in two categories: exergonic and endergonic reactions. Now, what do you mean by a exergonic or a endergonic reaction? As the name suggests, exer and ender, it relates with the uh, excretion and utilization, or internal and external. So exergonic reactions are those reactions which exert or which release the energy when they are catalyzed. Whereas the endergonic reaction is a reaction which utilizes energy to carry out itself. Okay. So in biochemistry, our cells, they carry out both the reactions. Basically, as far as the sustenance is concerned or cell proliferation is concerned, where a cell manufactures or a cell synthesizes some metabolites, it requires some amount of energy, it utilizes some amount of energy. So all those reactions which are referred to as biosynthetic reactions, where some sort of molecule is being synthesized in the cell. It may be a polysaccharide, it may be a protein, or it may be nucleic acid, whatever the cell synthesizes. A cell manufactures a chemical molecule inside itself. That reaction will be endergonic in nature. That reaction is going to use some amount of energy in the cell. Now, where from cell obtain this energy? The cell obtains this energy through the exergonic reaction, like catabolic reaction. Cell takes up a glucose molecule, breaks it down to carbon dioxide and water, and meanwhile, 38 molecules of ATP are being generated, and these 38 molecules of ATP are utilized. The energy from these ATP molecules utilized for the biosynthetic reaction. Okay, so there will be a sort of balance between a catabolic and anabolic reaction. You cannot say that cell will only carry out a catabolic reaction, or you cannot say that the cell will only survive when it carries out biosynthetic reaction. The cell has to carry out both the kind of reaction in a perfect balance, in a perfect rhythm, so that it not only sustains, it not only survives in a particular environment, but it should be able to proliferate, it should be able to multiply itself. Okay. Now, how this thing is possible, that energy is released from one reaction and it is utilized into the other reaction. What do you mean by energy? Now, if in physical terms, in our day-to-day life, if you talk about energy, what would be the energy? Energy would be heat, energy would be some kind of mechanical force or something like that. Okay. But when you are talking about a cell, what do you refer to when you say energy? Is it heat? Does cell generates heat? So in what form the cell utilizes the energy? We say in catabolic reaction energy is released, we say in anabolic reaction energy is utilized. So where from, when this energy is released, how it is stored, when this energy is utilized, how it is utilized. There should be some kind of storage form, or there should be some kind of intermediate entity which should store the energy released from the catabolic reaction and transfer it to the anabolic or biosynthetic reaction. For this purpose, cell utilizes various different kinds of chemical compounds or biochemicals to store this energy and to provide this energy into the latter phases. The most popular and the well-known molecule of such kind is adenosine triphosphate, or ATP, generally we call. If you remember, we call this ATP as currency of energy inside the cell. Means whatever the uptake and intake of energy is done, it is done mostly through ATP molecules, adenosine triphosphate molecules. Similarly, it is not so that the energy will be stored and provided through ATP molecules only. There are many other molecules also which can be utilized in a similar fashion. There are GTP molecules, guanosine triphosphate, there are uridine triphosphate, UTP molecules, there are cytidine triphosphate or CTP molecules. Apart from this, there is acetyl phosphate, there is diphosphoglyceric acid, there is phosphoenol pyruvic acid. These are some of the high energy molecules. We are seeing only 7 examples here. Why? It is not so that apart from these 7, no other energy rich molecule is available, but these are the most popular, most commonly involved, and high energy molecules. If you see in the next column, we have the kilocalorie per mole values of for these particular individual molecules. So for all these ATP, GTP, UTP, and CTP, you will see that the value here is 7.3 kilocalories. Means when one ATP molecule or any phosphate molecule is broken down, 7.3 kilocalories will be released, and that amount of energy will be used for some purpose. Similarly, we have other molecules like acetyl phosphate. It has got much higher energy as compared to ATP. It contains 10.1 kilocalories. When it is broken down, it releases 10.1 kilocalories. Or similarly, diphosphoglyceric acid is there, which releases 11.8 kilocalories, or PEP, phosphoenol pyruvate. This releases 14.8 kilocalories when it is broken down. Now, just remember, you should remember all these 7 molecules: adenosine triphosphate, guanosine triphosphate, uridine triphosphate, cytidine triphosphate, acetyl phosphate, diphosphoglyceric acid, or in short, we call it as DPA, diphosphoglyceric acid, and phosphoenol pyruvate or phosphoenol pyruvic acid. In short, we call it as PEP. These are all energy-rich compounds. These all act as an intermediate molecule for transfer of energy within the cell, and these are involved in various different biochemical pathways which we are going to study in some time. अभी हम आगे कुछ pathways study करेंगे, biochemical pathways. So you will be coming across many of these molecules. Just remember that these are energy transferring molecules. They may act as an energy transferring molecule during the biochemical reaction, from catabolic reactions to biosynthesis, and vice versa, biosynthesis to catabolic reactions. How it happens, we'll see in the pathways in detail. The next most important concept is the oxidation reduction reactions. Now, what these oxidation reduction reactions are redox reactions in short we call. Oxidation reactions, briefly you can define it as a reaction involving loss of electrons from the substrate molecule. And reduction is the vice versa case, gain in the electrons by the substrate molecules. Okay, so these are two different kinds of reactions. When this reaction occurs, suppose there is a substrate molecule, when it undergoes oxidation, means it it loses its electrons. Okay, so substrate loses the electrons, these electrons must be shifted somewhere else. Okay, they cannot be left as it is freely in the cell. So what happens? The enzyme removes the electrons from the substrate molecules. Those electrons, for time being, for temporarily, they are transferred to some other molecule. So when a substrate is oxidized, an intermediate is reduced. Why this thing happens? In substrate molecule, electron is removed, it gets oxidized, and whatever the electrons were being removed, those electrons, they are shifted to some intermediate compound or to some other chemical. So that chemical, since it is gaining the electrons, it becomes reduced. Okay. Vice versa, if the substrate is reduced, means a substrate is getting electrons from somewhere, some source. So after gaining the electrons, the substrate molecule becomes a reduced molecule. Whereas the source from which the substrate has taken those electrons, it becomes oxidized because it is losing the electrons. Okay, so oxidation reduction reactions in biochemistry, although chemically oxidation reaction is a separate kind of reaction, reduction reaction is a separate kind of reaction, but when you see the biochemical reactions occurring inside the cells, these reactions, they go side by side, oxidation as well as reduction, because if one molecule gets oxidized, other will be reduced. If one is reduced, other will be oxidized, and vice versa. This set of reaction will go side by side. So usually we refer them as oxidation reduction reaction. Okay. Now coming to the energy rich compounds, ATP, how ATP becomes an energy rich compound. So if you see the structure of these ATP molecules, so what ATP molecule basically is, that it is a phosphorylated, a highly phosphorylated form of adenosine. If you see this structure, this ribose sugar and this adenine, this dotted line, it depicts the adenosine. Okay, this much portion, this is the adenosine. Okay, this adenosine is phosphorylated one by various different kinds of enzymes through the phosphorus group. Means every time a phosphorus group is attached, adenosine becomes phosphorylated. Means this is adenosine molecule. Some kind of kinase molecule enzyme will act upon the adenosine molecule, and it will add up a phosphorus, so it becomes adenosine monophosphate, adenosine plus orthophosphate group, this group. Okay, this and this becomes adenosine monophosphate. Again, kinase enzyme acts upon it, adds up another phosphorus group, this. Now this becomes adenosine diphosphate. Adenosine monophosphate in short is called as AMP, adenosine monophosphate. When two phosphates are attached, it is called as adenosine diphosphate, ADP. Furthermore, this adenosine diphosphate is acted upon by kinase enzyme, and again phosphorylation occurs, and now this becomes adenosine triphosphate. Means there will be an adenosine molecule to which three phosphate groups will be attached. Okay. Now, how this molecule is energy rich? One thing regarding the phosphorus group, that this phosphorus group is a energy rich group. How it is an energy rich group? That whenever this phosphorus is broken down, suppose at this location it is broken down, so this phosphorus will be released. ATP molecule will become adenosine diphosphate. The adenosine will now have only two phosphorus groups attached to it, and when this bond is broken, 7.3 kilocalories of energy will be instantly released. Okay, this is how this ATP serves as a energy provider for the cell. Okay, whenever the cell is in need of energy, ATP will be acted upon by some enzyme. Phosphorus will be removed, and instantly 7.3 kilocalories of energy will be released, which will be utilized by the cell. Okay. Furthermore, one more thing, there will be a total or a continuous recycling of these ATP molecules. What happens that during the biosynthetic reaction, cell takes up these ATP molecules, breaks it down into ADP and generates the energy and utilizes that thing. Now, whatever the ADP molecules have been generated here, those ADP molecules, they then go to the catabolic pathways. The energy generating there, phosphorylation dephosphorylation reactions, when they occur in the catabolic pathways, phosphorus is removed from the substrate, and that phosphorus is taken up by this adenosine diphosphate, and it again becomes a adenosine triphosphate molecule. Means there will be continuous recycling, ATP being converted to ADP molecule, and again ADP being converted to ATP molecule. And in this way, the energy provisions are kept in check within the cell. The energy is simultaneously synthesized as well as it is utilized through these ATP molecules, or it may be GTP or UTP or any other energy rich molecule. But the energy storage and transfer will be like that. When there is low energy molecule, ADP is a low energy molecule as compared to ATP, so it will undergo phosphorylation. Phosphorus will come and attach with that ADP molecule. Now this will become adenosine triphosphate. When cell requires, it will take off that phosphorus, energy is released, and that will be utilized by the cell for its requirements. Okay. In this way, there will be continuous recycling. Now just consider that cell does not have ADP molecules. What is going to happen? Catabolic reactions occur. The energy is released during those catabolic reactions. But if there is no ADP molecule to receive that energy will be wasted. Therefore, these intermediate molecules are very much important, because it is not necessary that instantly catabolic reaction releases the energy and at the same time cell uses that thing. Even if that thing is there, it requires an intermediate molecule which could transfer this energy released during the catabolic reactions to the biosynthetic reaction. In that case, only energy will be consumed. Otherwise, if there is no intermediate molecule, there will be no utilization of the energy molecules. It would be totally wasted. Okay, this is the concept of energy transferred in the cell. Mostly in majority of the biochemical reactions, ATP serves as this intermediate molecule for transfer of energy from high energy to low energy reactions. Okay, but apart from this ATP, as we have seen those 7 examples, phosphoenol pyruvate is there, this DAP is there, or UTP is there, GTP is there. All those molecules may act as the source of energy or as the intermediate energy rich compounds during the biochemical reactions. Okay. Now, apart from the reactions or the inner concept of energy, there are various other concepts. Like we have said that most of the reactions are redox reactions. Okay, means what we say, the substrate gets oxidized, electrons are removed from the substrate, and they are taken by some other molecule. Okay, when a reduction reaction happens, the reverse thing happens. Means substrate uses or takes up electrons from some donor molecule and becomes reduced. Now, how this thing happens? For this purpose also, like ATP, what ATP is doing? It is acting as an intermediate molecule for transfer of energy, for transfer of phosphorus group. Okay. In the similar way, NAD acts as a intermediate molecule which acts as a acceptor of electrons. Now it has got two forms. As you could see here, This is the structure of your NAD molecule. Okay. Now, this particular portion, nicotinamide, nicotinamide, this particular part of your NAD molecule, this part is responsible for acceptance or donation of electron molecules. Okay. As you see here, this is the oxidized form. Now, what is the difference between this oxidized form and this reduced form? As you could see here, that this oxidized form, it lacks one hydrogen ion here, means one electron is less. Okay. Now, when some reaction, oxidation reaction occurs in which a substrate molecule is removed an electron from it. Suppose there is one oxidation reaction and electron is removed from the substrate molecule. That electron is accepted by this NAD molecule. Now, if you see here, this oxidized NAD molecule becomes reduced here. And these electrons, these hydrogen ions, they are being added up at this location. Okay. Now, this NAD becomes NADH2. Okay, NAD is the oxidized form. This NADH2 is the reduced form of the molecule. Now, what has happened? Oxidation reaction occurred. Hydrogen ion is being removed from the substrate. That hydrogen is taken up by this NAD molecule. It becomes reduced. The substrate has oxidized and NAD has reduced itself. NAD has become NADH2. Okay, or sometimes it is also written as NADH plus H, whatever. It becomes reduced. Now, why this is important, that there should be some electron acceptor molecule during the oxidation reactions, because on the first hand, you know, hydrogen is a very, very volatile substance. It is highly unstable. So what is going to happen, that when you remove hydrogen from the substrate, there are chances that will go back and reattach with the substrate molecule. There is one possibility. Another thing, that even if it remains freely suspended into the cytoplasm, that is going to adversely affect the electronic balance of the cell. It may alter the pH of the cytoplasm. And if such changes, either in electronic balance of the cell or in the pH of the cytoplasm, if such changes are to a higher extent, then they may prove detrimental for the survival of the cell itself. As you know very well, cells have their own optimum pH values. They grow, they live very well at a particular pH value. If pH fluctuates, their growth rates also vary depending on the pH. Okay, so if these hydrogen ions, they accumulate into the cytoplasm or into any liquid, what is going to happen? The pH is going to fall. It must become acidic. And if suppose the organism is a mesophilic organism, what is going to happen? When it is exposed to acidic conditions, it is going to die. Means, during the metabolic reactions, if this hydrogen is left free in the cytoplasm of the cell, it will become poisonous, it will become toxic, it will become detrimental for the survival of the cell, and cell will die. So this thing cannot be allowed. For this purpose, cell has got one provision. It has got electron acceptors like NAD. This NAD is there inside the cytoplasm. Whenever the hydrogen is removed or released from oxidation reaction, NAD accepts that thing. Now what next? At some extent, at some point of time, in another biochemical pathway, cell may require hydrogen. Cell may require electron to carry out reduction of some substrate molecule. At that time, the same NAD molecule goes, and here we have seen oxidized NAD becomes reduced, reverse reaction occurs. Means This reduced NAD is now oxidized. It sheds off this electron and it becomes oxidized. The electrons or hydrogen ions are released during this process of oxidation of NADH2 to NAD will be utilized and will be used to reduce the substrate molecule. Okay, so during the biochemical reactions, not only the physical conditions, not only a particular enzyme are important to carry out the reaction, but at the same time, such kind of intermediate molecules like ATP or GDP or GTP, whatever, or NAD or similar to this NAD, there are several other molecules also, like there are FADH2s, there are NADPH2s, which act in the similar manner. They act as oxidizing or reducing agents during the biochemical reaction, accept hydrogen from oxidation reactions and provide hydrogen to the reduction reactions. Okay, so these concepts you need to keep in mind when you are studying the biochemical pathways. In all the biochemical pathways, you will see that most of the reactions, especially those which are energy generating reactions, those which are energy consuming reactions, secondly, oxidoreduction reaction or oxidation reduction reactions, they require the involvement of either ATPs, energy का जहां पर मामला होगा ATP molecules होंगे. Where there is oxidation reduction reactions, there will be FADH2s, NADH2s, or NADPH2s, etc. Okay, so the role of these molecules will be that they will be acting as a transfer molecules. They are acting as a intermediaries during these reactions where some kind of product is being released. Okay, now the next most important thing, the synthesis of ATP. Now, if you see the ATP synthesis, so basically there are three ways of synthesizing the ATPs. One is the substrate level phosphorylation. Second is the electron transport phosphorylation. And third thing is photophosphorylation. So as far as this photophosphorylation is concerned, this is a process solely limited or restricted to photosynthetic organisms only. As the name suggests, photophosphorylation. Okay. First of all, phosphorylation means attachment or coupling of phosphorus. Okay. So phosphorylation means a reaction where phosphorus group is attached to the substrate molecule. Okay. Now we are talking about generation of ATP. Means basically what we are intending that ADP molecule is converted to ATP, means phosphorus is attached to ATP molecule and becomes a adenosine triphosphate molecule. So these things, they could be carried out in three ways. One is photophosphorylation, photosynthetic reactions. As we famously know that the plants can synthesize their own energy by way of photosynthesis. So all those reactions which are involved in this process, they are referred to as photophosphorylation, where an organism utilizes the daylight or sunlight to synthesize the energy or energy resulting compounds. Okay, so that will be photophosphorylation. Now, in other organisms, there are two ways. If you see the bacteria or if you see the fungi or like human beings, all the organisms, they may adopt either substrate level phosphorylation or they may adopt the oxidative phosphorylation, or which is famously known as electron transport phosphorylation, which is carried out through electrons transport chains. Now, what is the substrate level phosphorylation? SLP in short we call. We have an example here. We have 2-3 reactions here, as you could see here. This is one molecule 1,3-diphosphoglyceric acid being converted to 3-phosphoglyceric acid. Now, if you see here, what is this compound? 1,3-diphosphoglyceric acid means onto the glyceric acid at carbon number 1 and carbon number 3 means at this point and at this point, this is carbon number 1, this is carbon number 3, there are two phosphorus groups attached. So we call it as diphosphoglyceric acid, a glyceric acid which has two phosphorus groups attached to it. 1, 3 explains the position of the phosphorus group, carbon 1 and carbon 3. Now what is happening here, that this 1,3 diphosphoglyceric acid is converted to 3-phosphoglyceric acid. Means what is the difference between 1,3 diphosphoglyceric acid and 3-phosphoglyceric acid? That this 3-phosphoglyceric acid contains only one phosphorus. At phosphorus at this location has been removed. It is dephosphorylated. One phosphorus is removed from the carbon number 1. As a result, this 1,3 diphosphoglyceric acid becomes 3-phosphoglyceric acid. And during this, when this phosphorus is removed, this phosphorus couples with the ADP molecules and converts it to ATP. Okay. Similarly, we take another example, phosphoenol pyruvate. Now here you see this is one phosphorus attached to this phosphoenol pyruvate molecule. This is again dephosphorylated during the pathway. This phosphorus removed is accepted by the ADP molecule. It becomes ATP and resulting molecule is this pyruvic acid. On this location, the phosphorus is removed. यहाँ से आप phosphorus को remove कर देते हैं. Similarly, we see the third example, acetyl CoA. Okay. Now, if you see here, as such, you didn't see any phosphorus group attached here, but this acetyl CoA is a compound made with acetyl group and coenzyme ASH. Acetic acid and coenzyme A coupled together to make this acetyl CoA molecule. When it is dephosphorylated, when it is broken down, phosphorus is removed, it couples with the ADP and it becomes a ATP molecule, and resultant molecules are acetic acid and coenzyme ASH. Okay, so this is the substrate level phosphorylation carried out for synthesizing the ATPs. Similarly, there is another way of ATP synthesis called electron transport phosphorylation. That thing we'll see tomorrow. Okay, so for today, I think the time is up. We are running short of time. If you have any questions, any queries regarding this, you may raise your queries or we'll stop here.